evening. My name is Caitlin, and this is Willie, and we're your academic officers on the Graduate Student Association for Neuroscience. And it's our great pleasure to welcome you to the 2017 Integrated Program in Neuroscience 3-Minute Thesis Competition. So the 3-Minute Thesis Competition, or 3MT, originated back at the University of Queensland in 2008 and has since spread to over 200 universities across the world, including ours. So as you might have figured out, this competition challenges students to present their thesis research in only 3 minutes. Now this requires students to not only present their work in a way that's straightforward and accessible, but also engaging so that it can be understood by an audience, like you, from a diverse range of academic backgrounds. This not only requires students to be knowledgeable about their work, but also passionate and have excellent communication skills. This evening, we have 9 enthusiastic and talented IPN students presenting their work. I saw a dress rehearsal of these presentations on Tuesday, so I can assure you you are in for a great evening of presentations. At the end of the night, we'll be awarding a People's Choice Award, as well as the top 3 picks picked by our judges, who Willie will be introducing.
All right, thank you, Caitlin. So I will be introducing our judges, starting from ladies. So we have Dr. Tokumo Oyama. Dr. Oyama has a PhD in molecular and human genetics at Baylor College of Medicine. And she did her postdoc excuse me, at Dr. Marta Zaptik's lab. Sorry, it's very hard to pronounce. Genela Farm Research Campus and Howard Hughes Medical Institute. Currently, Dr. Oyama studies circuits, neurons, molecules, and genes that contribute to decision-making in fruit fly larvae. Then we have Dr. Austin Millerwood. Dr. Millerwood received his PhD in neurophysiology at the Open University. He worked at Dr. Leon Raymond's lab at the Department of Psychiatry and Brain Research Center at the UBC. Dr. Millerwood currently focuses on neural development, connectivity, transition during the early pathophysiology of adult onset diseases such as movement disorders and dementia. Moving on, we have Dr. Gary Armstrong. Dr. Armstrong had a PhD in biology at Queen's University. He took up a postdoctoral position with Dr. Pierre Drapeau at the University of Montreal. And And currently, his works focus on studying the pathophysiological association between specific genes, specific genetic mutations, and Lou disease by using larval zebrafish. And then we have Dr. Nicholas Sermakin. Dr. Sermakin had a PhD in biochemistry at the University of Montreal. And he worked his postdoctoral research at Dr. Paulo Sesson-Cosi, am I right? Okay, lab. And currently, he works on molecular mechanisms of circadian rhythms and the association with immunoresponse and psychiatric disorders in mice and humans. Last but not least, we have Dr. Wang Peng. Dr. Wang has his PhD at McGill in pharmacology, and he works his postdoctoral research at UBC. He focused on depression in animal models, specifically studying their physiology. Thank you for judges.
Let's get a round of applause for our judges. Now to the fun stuff. Our first presenter is Reinder Bostewil. He's a second-year PhD student at the MNI, and he will be presenting to us on gradients of hippocampal subfield connectivity and how they reflect long-axis specialization. So just so you know, everyone will have 3 minutes to present, after which they will be asked to stop. They know the cues that they'll be given. And also, if you could leave your questions until the end, we'll have a chance for all 9 participants to come up so you can ask them questions. Okay, and with that, let's get started.
Just maximize this one. tell you how you will remember me. Deep inside the brain is a structure called the hippocampus. And every single one of your hippocampi is currently working hard to encode a memory of this presentation. When you leave this building and you go home, you will need your hippocampus to retrieve the map of Montreal so you can navigate to your front door. In several common disorders such as Alzheimer's, epilepsy, and schizophrenia, we often find that the hippocampus is damaged. So this structure is of incredible importance in our daily functioning, or lack thereof. My research focuses on how does the hippocampus interact with the human brain, and how does behavior arise from these interactions. Now, we have a pretty good idea of how the hippocampus wires with the rest of the brain, but just knowing the wiring of a system doesn't actually tell you what it's doing. Consider, for example, this overhead projector over here. While its wiring is static, it always does the same thing. Depending on the input that we give it, this image behind me can change into pretty much anything. So knowing the wiring of a system doesn't tell you what it's doing, and the same holds true in the human brain. So how do we get at what the human brain is doing at any time? Well, when the human brain is active, the areas that are active require oxygen to produce energy, which they need for their activity. This oxygen is provided by blood. So blood flow toward an area is actually a good measure of the neural activity in that area. So using measures of blood flow, we looked at the neural activity in the hippocampus and several areas of the human brain, of the brain, in healthy adults. And what we've already found is that the front of the hippocampus tends to have the same type of activity as areas that are involved in more inward processes, such as memories of yourself or mind wandering. Whereas the back seems to have activity more similar to those in more current processes, such as your working memory. And that they have this similar activity means that they're communicating in some way, and thus likely means that the front is more involved with the same processes as the areas it communicates with. So, knowing what the hippocampus does in healthies can help us go back to these disorders, such as such as epilepsy. In one type of epilepsy, it's very common to remove the hippocampus as treatment to remove the epilepsy. However, this doesn't come without risks. You can lose functions following the surgery. If we have a better idea of what the hippocampus does, we can give these patients more information on what the surgery might do to them, giving them a better informed decision on what they'll lose. Now, I hope you'll remember my presentation, because then you will remember how you remember.
Thank you, Reinder. So next up we have Alice Bruneau. She's a first-year master's student at the Shriners Hospital, and she'll be talking to us about the evaluation of quantitative sensory testing as a clinical tool for the management of pediatric patients with chronic pain.
Hi, my name is Alice, and I have a question for you. Have you ever witnessed something more distressing than seeing a child in pain? I don't think I have, personally. Yet, pain in children is somewhat of a recent topic in medicine. In fact, up until the 1980s, surgeons performed painful interventions and surgeries in children and infants without anesthesia or proper pain management. Can you imagine? Fortunately, in the recent years, more attention has been brought to the fact that, yes, children do experience pain, and even chronic pain. We think it's a disease of the older population, right? Low back pain, joint pain, migraines, aren't these supposed to be for older people? Research shows that 1 Canadian adult in 5 suffers from chronic pain. Well, guess what? 1 in 5 children also suffer from chronic pain. And 2/3 of these go on to suffer from chronic pain into adulthood. Treatments are trial and error, and we give drugs to children without really knowing how they work in them. Only 3% of the children suffering from chronic pain get an effective treatment. 3%. The problem with pain is that it's subjective, right? We don't have a diagnosis tool for it, and except a numerical scale of 1 to 10, but my 2 out of 10 might be very different from your 2 out of 10, right? We don't have a diagnostic tool, we don't know how to evaluate it, and therefore we don't know how to treat it. And that's why only 3% get an effective treatment. The most important thing also with chronic pain is that some patients may have the same exact pain symptoms, but have very different underlying pathologies. For example, some patients may have an overload of their excitatory pathways that are responsible for pain transmission. For example, in the worst case scenario, just light touch would be interpreted by the spinal cord as a painful stimulation. It's a bit like when you have a sunburn and you try to put your shirt on, right? But try that 24/7 for 6 months. Other patients, on the other hand, might have a deficit in the descending pain inhibition, meaning their brains are not able to minimize pain like we would normally do. These two conditions are different, and they would respond differently to treatment because one involves the spinal cord and the other involves the brain itself. Now imagine for a minute if we had a simple diagnostic tool that would help us identify which mechanism is altered. Well, that's where I come in. I work on implementing quantitative sensory testing as a diagnostic tool for chronic pain, where we'd be able to identify any abnormality in the excitatory pathways or descending pathways. With this, with QST, hopefully a 9-year-old with a diagnosis of chronic pain would no longer be suffering for a lifetime. Thank you.
Okay, thank you, Alice. Next up, we have Arna Gosh. He is a first-year master's student working at the McConnell Engineering Building, and he'll be telling us about deep semantic architecture for the analysis of motor task EEG data. You don't have to if you don't. It would be live stream, so it might help.
Yeah. All right. Hello. Can everybody hear me? Yeah. Okay. The mic is good. Okay. So, are you going to show the time? So, hello everybody. I'm Arna, and I'm going to talk to you about deep semantic architecture for analysis of motor task EEG data. That was a mouthful. All right. But there are two important things you need to remember from this: deep semantic architecture and EEG data. So deep semantic architecture or deep neural networks or deep learning is a very powerful tool that can help you analyze and extract information from huge amount of data. I think you guys know about chatbots or the auto-tagging feature of Facebook. Well, it's all based on deep learning. So among the thousands of friends you have on Facebook, deep learning enables Facebook to find and tag those 3 friends you uploaded a photo on last night probably. So deep learning is also known as the artificial intelligence of today, and basically it is the intelligent eyes of Facebook. EEG or electroencephalography, as most of you would already know, it's an elegant and simple way to look at the brain activity. So you have these electrodes, which are basically sensors which can record brain activity, and they're just positioned on a cap. You wear the cap, turn it on, and ting, hello brain. All right, so why use the intelligence behind the eyes of Facebook to look at EEG data? Well, one, it helps improve the miserable lives of grad students working in neuroimaging. Because there's a lot of data and people try to do a lot of tedious data processing techniques to try and find out this needle in a haystack. Deep learning gives you those essential features, and then you can go and look at those features from the neuroscience perspective. Interesting, right? But a more interesting application, which has a greater impact than helping some dumb grad students, is to actually predict the efficacy of neuro-rehab techniques. Neuro-rehab techniques are what are used for the rehab of, let's say, stroke patients. So the exercise or artificial stimulation techniques, we don't really know how well each of these techniques works before we actually apply it on somebody who comes in who's a stroke survivor. Deep learning helps build a predictive model to do so. My work has been on using deep learning to actually try and investigate what exercise does to healthy subjects on a gripping task, as you can see here. So I have a model which can discriminate between subjects who did exercise and who did not do exercise before doing this gripping task, and from the EEG data. So I get essential features from the EEG data, and then I can look at what features, how did exercise change those features. Based on this information, we are looking to build a predictive model that can actually predict like what would the EEG data look like after somebody has done exercise. All right, just to summarize what I said for so long, it may be boring, but I believe there's a future where there's a deep learning assistant which can actually predict or which can actually find out the optimal neuro rehab strategy that can help stroke patients, whether it's exercise or, let's say, artificial stimulation techniques. And fingers crossed for improving the lives of neuroimaging grad students. Thank you.
Okay, thanks, Arna. Next up is Ben. And he is a fifth-year PhD student working at the MNI, and he'll be talking about musical predictions and pleasure. Why do our brains enjoy music?
Hi everyone, I'm Ben and I want to talk to you about why I think that music is imperative for our survival. So imagine for a second that you're in a Chinese forest. It's quiet, but you hear some soft thudding sounds. They're getting louder and faster. Is there a tiger charging at you? Our brains are always anticipating our environments to try to give us what we need and anticipate what is going to happen around us. When the environment isn't as we predict it to be, we need to be able to adjust quickly. We need to learn that tigers can be quiet too sometimes, and we need to start making new predictions like where that tiger is about to be. This is especially important when our survival is on the line, but we're making predictions all the time about all sorts of things. For example, even the cadence of my speech. Did I get you there? Maybe? So music is especially good at manipulating our expectations, at helping us to develop and test our predictions that we're always making. When our predictions are correct, that's great. Our brains are working well, and we keep those predictions so that we can use them in the future. When we're kind of right, but maybe not quite right, we need to adjust those predictions. So we hold on to them and refine them. And when we're not quite right, when our predictions are pretty far off, well, we're probably going to have to ditch those predictions and start from scratch. In music, we're constantly getting all sorts of feedback about our predictions. So as the music evolves, it's always establishing and violating patterns, and we're always able to predict and learn from the errors in our prediction as it goes on. So by this model, the music that would be best for us evolutionarily would be that which challenges our predictions a little bit, but not so much that we have to throw them out and start from scratch. So when I categorize music with a classifier according to its predictability, this is exactly what I find in the graph here. So the music that's medium predictable is that which people tend to like the most. This is a really strong and stable effect. So it seems that we like to learn from music. That's really cool. But where is this happening, and how does this happen for us? Well, when we learn about really evolutionarily important things like food or shelter, this blue area of the brain here, the nucleus accumbens, starts the learning process by quantifying exactly how much our predictions are off, the error in that prediction. So I designed an experiment in which people made musical predictions, and then I manipulated the outcomes of those predictions to see if this area did the same thing for music. It did, in the yellow part over there, which suggests that music is in fact evolutionarily valuable to us. So the people with more activity in this area also tended to learn more from their mistakes, which is a really important skill for all of us to do. So overall, music seems to feed a fundamental imperative for us of feeding our predictions so that we can learn from them. Thank you, that's my time.
Okay, thank you, Ben. Next up is Julia Santos. And she is a second-year PhD student who will be presenting to us on identifying molecules involved in neural identity and wiring.
Okay, who's gonna be doing the counting? You? Okay, so hello everyone. My name is Junia. I am interested in how innate behavior is formed. So raise your hands if you were taught how to breathe. Well, clearly nobody was taught how to breathe. And that's because breathing is an innate behavior. Things like riding a bicycle or speaking a language are not innate and they need to be learned. Our innate behavior is written in a code in our genes. So let's call it the molecular code. Many of these behaviors can be traced into connections in our brains. And when those connections are impaired, disorders such as autism arise. So to effectively help these patients, we need first to answer an essential question, which is, how do our neurons build connections to control our behavior? I approach this question by determining what is the molecular code for a neuron to build a circuit. I specifically look at the fruit fly, a single neuron in the fruit fly. This neuron is important for an innate behavior, which is related to its connections. So first, I start by deleting genes one by one in that neuron. Then I verify the changes in the connections and also in that reflex behavior. And to verify the precision of my code, I can take the same exact neuron from hundreds of animals and decode the instructions it has by sequencing. So combining all these results, I found out that 50 genes are required to wire up that circuit. The functions of those genes are the same across the animal kingdom. And some of those functions are like increasing the number of connections, decreasing the number of connections, or telling the wires where to go. So in summary, my results show that only a small number of genes is needed to drive innate behavior. By studying further those genes, we will be able to know how to reestablish the essential connections in the brain of a patient with autism. And maybe in the future, we will be able to add instructions to our code, and humans will be born knowing how to bike. Thank you.
Okay, thank you. And next up is Lauren Reynolds. Lauren is a sixth-year PhD student at the Douglas Mental Health University Institute, and she'll be talking to us about drugs of abuse and how they disrupt development of the adolescent prefrontal cortex.
All right. So, that was turned off. Okay, so teenagers are not known for making the best choices. Hi, my name is Lauren, and as an example, I want to say that luckily I was not stuck with my teenage choice of haircut forever. But some of the choices teenagers are making can have lifelong consequences. Drug use in adolescence increases the lifetime risk of developing and struggling with addiction. Addiction is a brain disease, and during adolescence, the brain is still under construction. So for us to know how to help people who begin taking drugs in adolescence, we need to know exactly what drugs are doing to the developing brain. But there is a problem. Until recently, we knew very little about how the brain was developing normally. Using a novel developmental application of circuit mapping techniques in mice, my thesis project showed for the first time that axons, the wires connecting different regions of the brain, are still growing to the prefrontal cortex during adolescence. The prefrontal cortex is a region of your brain that's really important for decision-making, planning, inhibition, and motivation, all types of behaviors that are impaired in individuals struggling with addiction. I also found that a protein called DCC is important in controlling the growth of these axons. DCC functions like a stoplight. It tells axons to either stay put in cortical regions or to keep growing into the prefrontal cortex during adolescence. Exposure to stimulant drugs of abuse in adolescence decrease the amount of DCC present in the brain. So if you've ever driven through a busy intersection that has a stoplight controlling the flow of traffic, you might be able to imagine the pandemonium when that signal malfunctions. By reducing the amount of DCC, drugs like amphetamine are essentially green lighting axons that should have stayed in subcortical regions to instead grow to the prefrontal cortex. This ectopic axon growth to the prefrontal cortex leads to deficits in those types of prefrontal cortex behaviors that we think increase the susceptibility to addiction. So what I'm interested in now is, can we prevent or restore, kind of rescue, the effects of adolescent exposure to drugs by increasing DCC signaling back to normal levels? So this information could be used to develop molecular interventions aimed at reducing addiction risk in people with a history of adolescent drug use and promoting healthy brain development. We hope that by implementing these treatments, future teenagers will be haunted for the rest of their life by their choice of prom date and not by their choices on drug use. Thank you.
Okay, thanks, Laura. And next up we have Nalima. She's a third-year PhD student at the Montreal Neurological Institute, and she'll be presenting to us on understanding the role of CURL3 in synaptic development and circuit formation.
Hello? Hello? Okay. So, from getting food all over your face as a baby to multitasking in your day-to-day life, how does this drastic change happen? Hi, I'm Neelima. Buckle up as I tell you more. The human brain has nearly 100 billion wire-like structures called neurons. These neurons make precise connections in the brain to help us process information and learn new things. Now, just like we need a cookbook sometimes to cook food, the neurons have a cookbook of their own, the DNA, which helps them make other molecules or proteins. Now, one such protein that might play a role to help neurons make connections is CURL3. Studies in mice have shown that removing CURL3 completely affects performance of these mice in tasks involving learning and memory. It's also known that these mice show defects in how neurons make connections in certain brain regions, such as the hippocampus, important for learning and memory. So we know that this protein is important here, but how is it important? We do know that CURL3 is a sticky protein. It recognizes other proteins like itself, and probably we think that this sticky property is important to help neurons form connections. The way I would test this is by using a technological tool called CRISPR. Now, CRISPR is a technique that makes use of a protein that functions like a pair of scissors would. It also makes use of another molecule that guides this scissor-like protein to target regions of the DNA to make specific changes that we want. Now, I want to make changes in CURL3 to disrupt the sticky property of the protein, and then go back to this system in mice and test if we see similar defects that were described earlier on when this protein was completely removed. Now, this will take us closer to understanding what the function of this protein is, and moreover, answer the bigger question of how neurons make connections in the brain. This will also help us gain insight into why certain individuals find it difficult to learn new things and make this ginormous switch from a baby to an adult. Thank you.
Okay, next up we have Scott Bell. He's a fourth-year PhD student at the Douglas, and he will be, I won't spoil it, but he'll be presenting on a new source of stem cells.
That's an optimization, really. Yeah. Do you mind if I now use the microphone?
Sure.
What if I told you that every day you're literally pissing science away? Hi, I'm Scott, and I work on making urine into stem cells, cells that can be used to be differentiated into virtually any other type of cell in the body. Now, I'm sure everyone here is familiar with some of the hype surrounding stem cells, that you can maybe use them to improve treatment, to better understand the human body, or to even extend our lifespan. And there is tremendous opportunities for stem cells to be used to improve our lives. But there are many obstacles that we need to overcome to get to that point. One big problem is how we acquire stem cells. In all of us, in me and you, there are cells that we can use to make stem cells. But we can't use just any cell. We have to use cells that can survive, divide, and thrive when we take them outside the human body. And the problem is that those cells tend to be located deep in our tissues, whether it's the pulp of our teeth or the bottom layers of our skin. And as a result, most stem cells today are generated from cells that can only be acquired if a surgeon goes and cuts a chunk of skin out of someone. That's expensive, it's invasive, it requires people to go to the hospital, it leaves a scar, and unsurprisingly, it is a very difficult thing to convince people to do sometimes, especially if they're children. So my fellow lab members and I thought that there could be a better way. So we went on a scavenger hunt, looking for better populations of cells that we can use, and where I was half scientist and half guinea pig. I had my skin sample, I had my blood drawn, I had my mouth swab, I had the roots of my hair pulled, and everywhere we looked, we found cells that were either too difficult to grow or too difficult to purify. Until I peed in a cup. You see, in every drop of urine, there's a few cells that have been scraped out of your kidneys or your bladder or the piping that connects them all. And these cells are exceptionally easy to isolate and purify. But they're not usually considered to be good cells to grow because they die very quickly in urine and they don't expand. However, I found that adding a few chemicals that help protect and nourish those cells extends the lifespan of them in urine from only a few hours to days. And that by mimicking the environment that these cells find in the bladder and kidney allowed them to grow just fine. This allows us to have a much cheaper, non-surgical, and effective way to generate stem cells. This protocol has been used to generate stem cells from patients all across Canada that never had to leave the comfort of their local clinic. If nothing else, I think it means that there are opportunities in science in what we throw away. Who knows what kind of scientific wealth is wasted every day? Thank you.
Okay, thanks, Scott. And last but not least, we have Sam Min. He's a first-year master's student at the Montreal General, and he'll be talking to us on the effect of patching duration on ocular dominance plasticity. I was looking for your first name. So I'm gonna use this microphone or use that one? One of them.
Hi, I'm Sam, but who we really should care about is Jack. This is Jack. He's 5 years old, he likes to smile and cry, and during the weekend, he would often go outside with his dad to play soccer. But he's not entirely normal. He has this condition called amblyopia, a brain developmental disorder that causes one eye to be strong and the other eye to be weak. So what is the treatment? Patching, which is exactly what it sounds like and looks like. So Jack's right eye is patched, and the right eye for Jack is the good eye. We should patch the good eye so that the weak eye could see more and improve over time. So the question is, how long should we patch Jack? For 6 to 8 hours per day. For how many days? Month to years. So during his entire childhood, from his school, 8 in the morning to 4 in the afternoon, his good eye would be patched. A lot of kids nowadays with a patch get bullied by their classmates just for wearing a patch. So what if I tell you that there's a shortcut to treating amblyopia? What if I tell you that we could treat the weak eye by patching the weak eye? So that's my project is about. My project is about patching the weak eye just for 2 hours for 1 day. And from testing normal adults like myself and my friends using psychophysical tools, we found that 2 hours of patching could bring about a huge improvement on the weak eye. So the next stage of this project is to transfer this research to amblyopic kids in the clinics and try to patch for 2 hours or 1 hour or 5 hours and see what would happen to the weak eye. So next time if you want to patch yourself, don't patch for 5 hours, don't patch for 4, just patch for 2 and you're good to go. Thank you very much.
Okay, so I think we have some time for some questions. So if actually the 9 contestants want to come back. So does anyone have... I see a question. So if anyone has a question, if you could just press the button in front of you, so that way everyone on the live stream can hear.
Can everyone hear me? So my question is for you, Nalima. Yeah. So you're using the CRISPR-Cas9 system. Have you already generated those mouse knockouts, or sorry, those mouse mutations, the indels?
So at the moment I'm at the stage of I've designed the guides and I'm testing how efficient these guides are, and hopefully once we confirm that we're in the process of generating the knock-in.
Okay, yeah. So my second follow-up question is, do you have a certain size of a deletion you want to make in that protein, or
yes.
Are you going to be trying to have homologous recombination to knock in just like, I guess, a stretch of DNA that's not going to disrupt it?
So we, from the information we have from our collaborators who are looking at this protein from a non-biological context, we know that a point mutation in this protein is involved in this Chiral 3 homophilic cell adhesion. So by introducing that point mutation, we want to disrupt this property in mice.
Thank you.
Thanks.
I have a question about the amblyopia. So I assume the line of logic behind patching the strong eye is just allowing the weak eye more sensitive and hopefully strengthening its circuits. But I'm wondering, what's the logic behind patching the weaker eye?
So there's this theory, gain theory, where the dominant eye inhibits the input to the weak eye, and the weak eye inhibits the strong eye's input, and that occurs reciprocally. And that through that theory, we think that the weak eye, when it gets patched, it starts to adapt when there's no visual input. So during the patching state, it gets more sensitive. It's like a homeostatic mechanism, and that's called gain theory.
Thank you.
Anything else?
Question for everyone. Has anybody seen a presentation that wasn't theirs that made them want to switch what they were doing?
I have a question for Arna. Hi. So what sort of features do you look at in your EEG data? Do you just process like the whole raw data, or do you look at certain ERPs?
Yeah, that's the magic with deep learning. You don't need to give features, it extracts features. And for me, I give time frequency maps, if that answers your question. Like, it's not raw time signal data, it's time frequency data.
So now we're gonna take a break so we can decide who the big winners are. So if you all would like to go get some refreshment.
Hold on. So while the judges are deliberating, we will have you to vote for your preferred presentation and elect the people's choice. I just need to get the link on the screen.
No, the refreshments are there, so you guys can stay seated, we'll go out.
As you like.
be judge next year.
Outside, where the judges are.
All these questions.